Okay. So winners, let me take a complete review of last discussion over the cell cycle. We started with the cell cycle. Since the main phases of cell cycle is the birth of the cell, the development or growth of the cell, and then its death of the cell. Definitely in terms of unicellular organisms, if we talk about the unicellular organism as compared to multicellular organism, so unicellular organisms ka growth ही reproduction. In fact, in terms of growth, if someone will start discussion, then only the reproduction will come in our mind. Because if the cell is growing, then definitely it has to be divided. So वो growth और division का एक aspect देखते हुए cell cycle की दो phases बनाएंगे. No problem. Lisa, I'll try my best to upload the lecture. Usually मैं इसलिए upload नहीं करता हूँ same day, so that बच्चे attend करें classes. I'll upload it, no problem. Coming back to the discussion, so cell cycle is basically focused on cell division, because cell division is itself cell give birth to another cell, or we should say the daughter cells or the progeny cells. It grows and again it divides. So in context to cell cycle, we have to deal with cell division only. That cell cycle consists of two phases. Anyone? Instant answer.
Interphase.
One is interphase and one is M phase. Is it?
Yes, sir.
Interphase and M phase. Interphase, which is a resting phase as far as the division is concerned. Because in interphase, we do not expect the cell will divide at all. But yes, cell will grow itself or prepare itself to at least divide. So interphase is nothing other than a resting phase. But the most important question that is asked, that is frequently asked question in all the competitive exam is, the interphase is resting in respect to what? Whether it is metabolically active? Yes. Metabolically most active phase is interphase. Yes, we do agree. And the rest, the another one is the M phase. M phase is mitotic phase. Mitotic phase is the divisional phase, actually the phase of division, where the cell divides into two. So there are two stages, one is interphase and one is M phase. That interphase and M, am I audible?
Yes, sir.
Power cut हुआ बच्चों, इसलिए light का effect थोड़ा सा change हुआ होगा, but don't worry. So interphase has three phases, G1, S, G2. I hope we had discussed but not write anything about G1, S, G2. Is it?
Yes, sir.
Yes, sir.
कौशिकी मैडम हमने पढ़ा है जरूर, but हमने शायद उसको लिखा नहीं था. G1, S, G2. G1 is growth 1, or I should say the post-mitotic gap phase. S is the phase of synthesis, that is DNA synthesis. And G2 is the growth second, or I should say the pre-mitotic gap phase. I hope we had discussed these 3 points. Is it?
Yes, sir.
Yes, sir.
Perfect. We'll write in details, no problem at all. G1, S and G2. Growth 1, which is known as post-mitotic gap phase. S phase, this is phase of synthesis of DNA, not the chromosome. Not the chromosome. And G2 is the growth phase, second, or I should say the pre-mitotic gap phase. So G1, S, G2 and M phase. G1, S, G2M, G1, S, G2M, G1, S, G2M. This is the basic cycle. And I had discussed, I hope I told you about the 2 checkpoints. Anyone can tell me those 2 checkpoints?
CM and CG1.
Perfect. This is G1. Sorry, this is G1. This is S. This is G2 and this is M. M has 4 phases. Anyone? Anyone?
Prophase, metaphase, anaphase.
Metaphase, anaphase, telophase.
Prophase, metaphase, anaphase, and telophase. Definitely these 4 are the subphases of this M phase. Perfect. And there are 2 checkpoints. There are 2 checkpoints. One checkpoint is what? CG1. The another checkpoint is what? CM.
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 Mitotic cycline and growth of one cycline. And this cycline is cycline dependent kinase basically. CDK, cycline dependent kinase. एक cycline, CDK cycline, and one is cycline G1. So these two are the checkpoints. These two checkpoints check whether the cell is really have sufficient energy or enough energy to divide, or secondly, whether this cell has sufficiently grown up so that it can enter into karyokinesis. Because this M phase consists of two subphases. What was this?
Karyokinesis and cytokinesis.
One is what? Karyokinesis and
Cytokinesis.
cytokinesis. So karyokinesis, what is karyokinesis? What karyo means?
Nucleus.
Nucleus.
And what is cytokinesis?
Cytoplasm.
Cytoplasm division.
बहुत बढ़िया. So ये सारा discussion we had done with this. Only the point we have to write what are the characteristic or particular feature of G1, what are the features of S, and what are the features of G2. G1, S, G2, and then one by one the phases. But before that, we already also discussed about the mitogens. Anyone? The mitogens and mitotic poisons? Anyone?
Factors
So
that
mitosis
बोलो बोलो कोई भी बोलो. No
factors
problem.
that promote cell division.
Mitogens that promote cell division, right? Or
Mitotic
Mitotic poison
inhibit cell
inhibits.
division.
inhibit cell division. ये तो बच्चा भी बताएगा दोस्तों. तुम तो विनर हो. कुछ और बताओ. Mitotic poison का कोई नाम बताओ. Give some example of mitotic poison.
Radiation.
Radiations. Radiation क्या लगता है आपको? रोक देता है radiations? Cell division को promote करता है कि रोक
No
देता
sir,
है?
promote करता है.
Promote करता.
मैंने उस दिन भी आपको बताया था, फिर रिपीट कर रहा हूं दोस्तों. रेडिएशन थेरेपी दी जाती है, रेडियोथेरेपी होती है, कीमोथेरेपी, बट दोज रेडिएशंस आर ड्रग इंड्यूस्ड. अंडरस्टूड विनर्स? यस. सिद्धि मैडम स्पेलिंग गलत है. दैट इज नॉट कैल्चिसिन, दैट इज कोल्चिसिन. सिद्धि, दिस इज व्हाट? कोल्चिसिन. This is colchicine, which is a big mitotic poison, which is frequently asked question. This is colchicine. What colchicine does? Colchicine inhibit the formation of spindle fibers. स्पिंडल फाइबर के फॉर्मेशन को जो रोक देता है वो कोल्चिसिन. अब जब स्पिंडल फाइबर ही नहीं बनेंगे, so no one can expect the chromosome and chromatid will get separated. Perfect. So these are the some basic ideas. Let us start with. Let me check your NCERT. Okay. Let us start with the interface. हेडिंग डालिए सेल साइकिल. कुछ बेसिक्स हम लोगों ने लिख लिए थे बच्चों के, नहीं लिखे थे? विनर्स, अरे कुछ लिखा था, भूल ही नहीं रहा यार. प्रोग्राम्ड cyclic changes in a cell is what? Cell cycle. Perfect. In which what we consider in this, we consider birth to death. Do you know there are two types of death? Do you know?
No, sir.
No sir.
No, sir.
What you know यार, क्या ही कर रहे हो विनर्स? कॉमन थोड़ा सा पढ़ते रहा करो. प्रोग्राम सेल डेथ एंड सडन डेथ. प्रोग्राम सेल डेथ, ये तो ऐसे ही आ गया दिमाग में इसलिए मैंने आपको बता दिया, नॉट नीडेड हियर. प्रोग्राम सेल डेथ, सेल का बर्थ, सेल का ग्रोथ, देन सेल का डेथ, इट इज ऑल प्रोग्राम्ड बाय अ जेनेटिक मशीनरी. This genetic machinery is such a brilliant machinery that a cell try their best to follow those instructions, and rarely due to some mutagens due to some oncogenes, this cell cycle or cell reproductive pattern changes abnormally or abruptly. Otherwise, cell always works, functions in a programmed manner. And this death, such a death is known as programmed death, or I should say apoptosis. What is the word? Apoptosis. Apoptosis. Apoptosis is programmed cell death. And unprogrammed means sudden death due to some injury, due to some accidental part, due to some drug reaction, whatever the reason is. If suddenly cell dies, then this is known as necrosis. And this is known as necrosis. Okay. So just for your knowledge, I shared these two terms with you. Coming back to the discussion. So in this, we consider the birth to death and the duration between two cell cycles. The duration between two cell cycle is generation time. दो सेल साइकिल के बीच का समय जो पीरियड जो है बच्चों, that is called as generation time, which may definitely vary individual to individual or not individual to individual, species to species. Yeah. If human cell, then definitely सभी का सेल उसी तरह से डिवाइड होगा. If it is of cattle, अलग तरीके से होगा. If it is crop plants, plant cells, अलग तरीके से होगा. So generation time is the time between two cell cycles. Okay. It involves two stages. One is interphase and one is M phase. So let us start with interphase. Let us start with interphase. Interphase में समझा तो सब दिया, लिखने का बात रहेगा आपका. I have shared a cycle with you, is it?
Yes, sir.
Okay. So have you noted it down, this cycle, so that I should not repeat it again? You are noted or not? Tell me.
Noted, sir.
Noted. चलिए तो कुछ पॉइंट सीधे सीधे, नो प्रॉब्लम, ज्यादा डिटेल लिखने की जरूरत नहीं. इंटरफेस में स्टार्ट करते हैं. जीवन, कुछ स्पेशल पॉइंट्स जीवन के मैं आपको बता दूं. दैट ओनली वी विल राइट. द जीवन इज also known as what? Jivan is also known as what?
Post mitotic phase.
Growth.
Post mitotic gap phase. Understood? Understood?
Yes, sir. Yes, sir.
Perfect. Synthesis of RNA, protein, synthesis of RNA, protein, enzymes, histones, etc. Noted?
Yes, sir.
Perfect. Organelles also increase in numbers. G1. At G1, cell has three options. सेल के पास तीन ऑप्शंस. क्या तीन ऑप्शंस हैं वो बता दूं. ये शायद मैंने एक्सप्लेन भी किया होगा. इज इट?
Yes, sir.
बता दिया था ना. चलिए कोई बात. पहला कंटिन्यू, जीवन से आगे कंटिन्यू जाए और एस में एंटर कर जाए. दूसरा डिसकंटिन्यू और G0 में चला जाए. और तीसरा अरेस्ट और G1 में ही पड़े रहे. there are three options, simple three options are there. If the cell finds itself that sufficient energy is not available, no enough potential is available, so there are three choices. If energy level is not sufficient, then permanently it can enter into G0 and can exit from the cell cycle. Once it enters into G1 and then come back to G0 and exit, then there is no chance for further reproduction. Now the cell is exited or exit from the cell cycle. And those cells which enters into G0 permanently, those differentiate there and never ever divides once they form. And such examples are nerve cell and muscle cell. They permanently enter into G0 and then never ever divides. Or in case of plants, we should say the permanent tissues. Because until unless, or I should say as long as, not until unless, as long as they are meristematic, they will keep on dividing, keep on dividing, keep on growing, keep on dividing. But they are not differentiating themselves. Once they enter into G0, because of whatever the reason is, once they enter into G0, they stop division. And once they stop division, they start differentiating. So G0 is characterized by differentiation and hiring a function of a cell. As long as we were a zygote in the womb of our mother, as far as a seed is in dormant state, seed in dormant state is metabolically suspended cell. This is arrested one. वो dormant हो गए, metabolism suspend कर दिया. But it can grow into root, into shoot, into leaves, into flowers. So it is keep on dividing once it is get activated or once get the favorable conditions. This is meristematic tissues. In case of human beings, we never use the term meristem, but the cell which is keep on dividing is not yet hired a specific function or not at all differentiated. Once any of the cell, whether it is plant or animal, if it differentiates, it has to hire a specific function. When we were embryo, the single cell was capable to divide and to produce ears, eyes, nose, lips, hair, complete body, liver, kidney, heart. But once it becomes heart, it stops further division because it is differentiated. And once it differentiated, it has to hire a specific function. Understood? G1, G0 concept understood?
Yes, sir.
Perfect. Perfect. So either it should continue if energy is sufficient, either it can discontinue or can arrest, can take a pause. Or I should say that every cell has to enter into G0 at least once. But if enters into G1, decide to continue and again back to G0. I should not say again back to G0. वो G0 G1 का ही पार्ट है. तो G1 में आने के बाद या तो वहां रुक जाए, अरेस्ट हो जाए, या तो आगे बढ़ जाए, या तो भैया परमानेंट G0 में चला गया. तो जो 0 में चला गया, that will be exit and will hire a specific function and will differentiate. But हम बात कर रहे हैं आगे बढ़ने. So we should continue with G1. Okay, we should continue with S. So the second phase is S phase. S phase is known as phase of DNA synthesis. Phase of DNA synthesis, that means the haploid content become diploid, or haploid diploid I should not say, because haploidy and diploidy is the term used for chromosomes and I'm talking about the DNA. I hope we have already discussed enough for a chromosome chromatid gene, is it?
Yes, sir.
But
Yes.
Yes, sir.
still no problem, I can repeat. This is basically a chromosome, which was earlierly a thin chromatin thread This was a thin chromatin thread, which is their normal position, which is a normal status of any cell, which is not at all dividing. But once it decide to divide, then it has to enter into G1S, G2. So when it enters into S, its contained become doubles. Thread never. In fact, मैंने आपसे कहा था क्रोमोसोम जैसी कोई चीज नहीं होती दोस्तों. Chroma, colorful zoma body. That colorful body is observed in the microscope and it's visible, so it is called as chromosome. Otherwise, it is nothing other than that chromatin thread. But since it decides to divide, it has to enter into S. Once it enters into S, the content must be doubled. So DNA content becomes doubled, 2C to 4C. And when it becomes 4C, how this thin thread remains thin? It becomes thicky and appears like two sister chromatids. Understood? Understood?
Yes, sir.
So
Yes, sir.
if Siddesh bhai, if there is a gene for height, capital H, then here must be capital H. Yes or no?
Yes, sir.
Because it's a copy, na? It's a replica. So that's why what I'm saying, the phase of DNA synthesis, or I should say, called as phase of DNA replication. I hope you are getting my points, winners.
Yes, sir.
Perfect. So that DNA becomes double, 2C to 4C, the DNA content becomes double, and such body observed, such observed bodies, seen body are known as chromosome and found to be 23 pairs in human beings, out of which, how many are autosomes?
22 pairs.
22.
So I should say 22 into 2 means pair and 1 into 2 means one pair. These 44 are autosomes which are absolutely same, or absolutely I should not say. These are same in case of male and female because it represents human beings. And what differentiate male and female is XX or XY. So 44 plus 45 plus 1, 46. Either this 46 or this 46. So I should say the 23 pairs of chromosomes present in human cell. And I hope now you understood. These two chromatids are known as sister chromatids. Agree?
Yes, sir.
And Dr. Vyavhare, I should not say Dr. Vyavhare, Binner Vyavhare, Ishan Mohan, Ishani Nikhade, Kaushiki Jha, Mandar Deshmukh, बताइए मुझे, is it sister chromatids? Yes or no?
Yes, sir.
आवाज भी कैसी आ रही बिल्कुल यस सर
Yes,
जैसे जोरदार बोलो यार.
Yes, sir.
These are what, Kaushiki? These are sister chromatids. And there is a point in between. क्या कहा था उसे मैंने?
Centromere.
Perfect. This is centromere, but one more structure was there.
Kinetocosis.
Kinetosis.
Smira madam, what was the relation between centromere, kinetochore, chromosome and chromatid?
One chromosome is equal to 2 chromatid is equal to 2 kinetochores is equal to 1 centromere.
Chromatid, 2 kinetochore and 1 centromere. Perfect. So now good to go. The next one is, if we talk about the S phase, definitely I should say invisible phase bhi kehlate because chromosome prepare themselves for equal distribution. No problem. इससे ज्यादा इसमें कुछ बताने की जरूरत नहीं मुझे. नोट जरूर मैं लिख सकता हूं. DNA gets double, not chromosomes. I should go for G2 phase.
Excuse me, sir.
G2 phase. Namisha, Parth, Pranav, are you there?
Yes, sir.
Yes, sir.
G2 phase definitely उसमें growth हो चुका है सारा, है ना? इसे second growth phase भी कह देते हैं. सेकेंड ग्रोथ फेज, और एक क्या नाम दे दूं इसे मैं?
प्री माइटोटिक।
Pre-mitotic.
प्री माइटोटिक गैप फेज। परफेक्ट। चलिए, सो second checkpoint here, note, let me check, एनसीईआरटी में तो आपको G2 में कुछ और तो क्या ही लिखाया गया होगा, ट्यूबुलिन प्रोटीन, बस, and that's it. Cell undergoes the second checkpoint. What is this?
CM.
CM. Perfect. Now, since we want to study the cell division, so we will continue with S phase. We continued with this. G1S and G2 is done. G1S and G2 is done. Now we need to start what? Mitotic phase. Agree? But
Yes, sir.
before that, I should write a few points for G0 because it is as good as important for competitive exams, क्योंकि बच्चे इसको इग्नोर कर देता है ना. Inactivation of cell. Inactivation of cell. Just a minute. Okay. Inactivation of cell cycle due to unavailability of, I should say, the mitogens. Unavailability of the mitogens. And energy also. And energy. Though cell remain metabolically active. ये इंपॉर्टेंट पॉइंट है जो कंपटीशन में पूछा जाता है कि सेल रिमेन मेटाबॉलिकली एक्टिव. It may exit and enter into the phase of differentiation. यदि ये डिफरेंशिएशन हो गई तो इसको कहते हैं exit. If not differentiated, then no problem. It will continue to grow or continue to divide. Should I start with mitosis? Yes, Om, raise your hands. Om.
सर वो आपने बोला था कि मतलब
Om, you are not audible.
सर आपने बोला था कि जो क्रोमेटिन मटेरियल
Om, you are not audible. तू या तो माइक पास में कर या साउंड बढ़ा.
सर माइक भी पास में है और साउंड भी फुल है.
अरे यार. Anyone can hear Om?
Yes, sir.
हाँ Om बोलो.
सर आपने बोला था कि क्रोमेटिन मटेरियल का कंटेंट डबल नहीं होता.
एक मिनट ओम, या तो मुझे कुछ करना पड़ेगा क्योंकि बाकी सबकी आवाज आ रही है ओम यार, आपकी आवाज नहीं आ रही है मुझे. जस्ट मिनट, क्या करना पड़ेगा मुझे?
सर आपको
Anyone have-
ईयरफोन या फिर हेडफोन लगा के सुनना पड़ेगा उसको.
मैं टाइप कर देता हूँ.
वो टाइप कर रहे हैं सर.
अच्छा ओके. ओम फिर भी कोशिश कर, तू बोल क्योंकि टाइम लगेगा.
सर वो आपने बोला था कि क्रोमेटिन मटेरियल का कंटेंट इंक्रीज होता है लेकिन नंबर डबल नहीं होता, और एस फेज में डीएनए का नंबर बढ़ता है, तो क्रोमेटिन में तो मतलब वो हाईली कॉइल्ड डीएनए ही रहता है ना, तो फिर ये तो दोनों कॉन्ट्राडिक्ट.
अच्छा ओके ओके ओके. नहीं, आपने थोड़ा सा कंसेप्ट गलत पढ़ा. ओम, थोड़ा सा डॉक्टर गलत पकड़ा आपने. गलत कैसे पकड़ा? अच्छा, बैटरी भी डाउन हो गई है मेरी, ऐसा. जस्ट मिनट. ठीक, छोड़िए. ओम, आपने कंसेप्ट थोड़ा सा गलत पकड़ा बच्चे. गलत ऐसे पकड़ा कि मैंने आपसे कहा डीएनए कंटेंट्स गेट्स डबल. जब डीएनए कंटेंट डबल हो रहा है तो ये पूरा थ्रेड डीएनए ही तो है. मैंने आपसे कहा वन क्रोमोसोम कंसिस्ट ऑफ टू क्रोमेटिड्स. दिस क्रोमेटिड कंसिस्ट ऑफ क्रोमोनिमेटा. This chromosome contains chromonimeta. That chromonimeta is chromatin actually, and this chromatin is actually nothing other than the DNA. So their contents are getting double and accumulated. Threads are not doubled. Om,
यस सर.
भाई मैं आपसे कहूं ये पेन है. अभी ओम कितने पेन दिख रहे हैं आपको?
1.
ओम, how many pens are there?
सर 1.
एक. अभी
सर 2.
दो यार तुझे दिख रहे हैं. अभी तो एक ही तो दिख रहा है तुझे यार. ओम, अभी
हाँ मतलब साइड एंगल से 1 दिख रहा है.
हाँ, साइड एंगल से काय को सीधे ही देखना मेरे भाई. तो जो एक दिख रहा है लेकिन एक थोड़ा थिक दिख रहा होगा ना,
हाँ सर.
तो थ्रेड नहीं बढ़े, उसका कंटेंट डबल हो गया. अंडरस्टूड?
यस सर.
Perfect. Should I start for M-phase? Please check. If any doubt, tell me. Anyone? Achal, Arnav, Ishan, Parth, Pranav, Siddesh,
No, sir.
Prathamesh, Mugdha, Yash, Anirudh, Urvi, Tejas, Soham, Smira, Siddhi,
Yes, sir. Yes,
Shaunak,
sir.
Dahibate, Shreya, Kadu. Any doubt? Chali. I think I should go for mitosis. And let me very clear at one front. If I'm going to start mitosis, let me clear it at one go. Theoretically तो बाद में देखेंगे, लिखेंगे, but let me very clear at one go. Since I'm talking about prophase, metaphase, anaphase and telophase, basic पकड़िए बच्चों, पेन डाउन कर दीजिए और पूरा फोकस लगा दीजिए इस पर. Metaphase, anaphase and telophase. See what I'm telling you. Since we are already, a cell already passed from S phase, so we expect the DNA contents is already doubled. Do you agree with this?
Yes, sir.
So, what we expect that chromatin material become thicky. Do you agree with this?
Yes, sir.
Yes, sir.
Yes.
So that thicky material, if I'm saying you, here 23 pairs of chromosomes is accommodating in this, I should say this 23 pairs of chromosomes is in this pocket, suppose, or in this area. So what do you think? If it becomes 46 pairs, so it does require more space to accommodate, yes or no?
Yes.
Yes, sir.
भाई जाहिर सी बात है यार, मैंने जो कपड़े पहन रखे हैं थोड़ी देर के लिए समझो कि ये मेरी मेम्ब्रेन है. I am not getting doubled. Nitin Pathak is not getting doubled. But the content inside my body is getting doubled. The material that will form 2 more ears are doubled. The material or cartilages that will form 1 more nose will get doubled. So everything will be thickened now. Yes or no?
Yes, sir.
Yes, sir.
My size will be a very beautiful size. What do you think? Yes or no?
Yes, sir.
Yes or no? So if I'm not getting double, but my contents become double, what do you think? This cloth or this shirt will start rupturing? Yes or no?
Yes,
Yes,
sir.
sir.
Yes, sir.
Perfect. बस यही मैं बताना चाह रहा था आपको. That's why I'm not preparing this as cell. I'm preparing this circle as a nucleus. As a nucleus. Cell तो बहुत बड़ी है. मैं cell के nucleus की बात कर रहा हूँ. Because I'm talking about what?
Karyokinesis.
Karyokinesis. So I should discuss with karyo only, with the nucleus only. So first of all, if I'm talking about the prophase, the first phase, which is coming after the getting duplicated content, and nucleus is not ready to accommodate at all. So it has to get rupture and disappear. What do you think? Yes or no?
Yes, sir.
Yes, sir.
So inside this, what I expect, the first phase, मैं theory लिखा दूंगा आपको. That is not a big issue. Just try to understand. Whatever contents become double, those appears like chromosomes. This appears like a chromosome. Since it doubles, that only it can be seen. Otherwise तो देखा ही नहीं जा सकता था ना. देखता तो केवल chromatin thread है. वो देखा ही जा रहा है इसलिए because वो thick हो चुके हैं. So I expect कि such if it is a human cell, then I expect कि such a pair 23 pairs will be like this. Agree, winners?
Yes, sir.
Perfect. So first of all, if someone will ask you what are the characteristic feature of prophase, winner should not or proud resonant must not be afraid of the question. Simple. Since it is coming after the S phase, content getting doubled, not the thread. So it is tough for the nucleus to accommodate such a big content. But still it tries to pressurize that material and material gets condensed. So the prophase, first character of prophase is condensation of the chromatin material. Condensation of the chromatin material. But it is still, the membrane has to disappear because such a double content can't be accommodated in a single nucleus. Since all nucleocytoplasmic ratio is disturbed, so nuclear membrane has to disappear. Character number 2. What was the character number 1? What was the character number 1 of prophase?
Condensation of chromatin material.
Condensation of the chromatin material. Number 2 is
The membrane disappears.
disappearing, start disappearing the nuclear membrane. One more. Since now the nucleuses keep on expanding, so some organelles must to be disappeared. Yes or no?
Yes, sir.
Yes, sir.
क्योंकि लॉजिकली पकड़ना, लॉजिकली कंसेप्ट बताना मेरा काम है. थियरी तो मोर और लर्न, थोड़ा सा डेटा इधर उधर लर्न करना पड़ेगा, फैक्ट्स लर्न करने पड़ेंगे. बट फैक्ट ये है, कंसेप्ट ये है कि आपने कंटेंट डबल कर लिया है तो उसको कंडेंस्ड होना पड़ेगा. स्टार्ट कंडेंसेशन, न्यूक्लियर मेम्ब्रेन डिसअपियर्ड, सम ऑर्गन्स स्टार्ट डिसअपियरिंग. दैट्स इट. दिस इज द प्रोफेज. When it comes to metaphase, all those 23 pairs, I should say, the 46 chromosomes aligned on equator plate. This is equator plate. All chromosomes aligned on equator plate, and G1 S, G2 में अपन ने लिखा नहीं, G1 में लिखना था centriole formation starts. Centriole formation starts. जो S phase में continue हो जाता है. Centriole is the part of centrosome that moves on opposite pole. Prophase को we can divide into early prophase, mid prophase, late prophase, and finally metaphase में there are some spindle fiber that attach to kinetochore. Agree? Agree?
Yes, sir.
आवाज नहीं आ रही है. Agree?
Yes, sir.
Yes, sir.
6 number of chromosomes aligned on a single metaphasic plate. Why I'm emphasizing this word, single metaphasic plate? Because when I'll teach you meiosis, I'll use both the terms. In meiosis 1, I'll use double metaphasic plate. And in meiosis 2, I will use single metaphasic plate. Only on equator, single metaphase plate is there. 46 chromosomes aligned on equator, and the spindle fiber that arose from the centriole joins to kinetochore. There are three types of fibers, my dear. Sab likhenge detail. There are three types of fibers. One is continuous fiber, one is discontinuous fiber, and one is chromosomal fiber. One is chromosomal fiber. Continuous fiber continue from one pole to another pole without binding to chromosome. The discontinuous fiber may come to half and may come to half, not again binding to any chromosome. But the chromosomal fiber joins at kinetochore. Understood? Understood?
Yes, sir.
Yes, sir.
आवाज भी नहीं आ रही तुम लोगों की यार. चलिए. Mr. Dahi Bhate, are you there? Shreya madam, Siddhi madam, Manoli madam, Urvi,
Yes, sir.
Yes, sir.
Parth, Bishraji, Namisha.
Yes, sir.
Yes, sir.
So when it comes to the equator plate, this is called as metaphase. When it was condensing, disappearing, organelle disappearing, then it was prophase, which can be divided into early, pre, and yes, Mira, you raised your hands.
Sir, what happens to the nuclear membrane in metaphase?
Still it has disappeared, completely disappeared. That's why, Smira, try to understand. It's a good question. What do you think? Centrosome lies inside the nucleus or outside the nucleus?
Outside the nucleus.
फिर कैसे मैंने यहाँ पर जोड़ दिया? That means what? Nuclear membrane completely disappeared. Agree?
Yes, sir.
That's why the centriole comes in contact with the chromosome, na? Otherwise, one material is outside the nucleus, one is inside the nucleus. How these two are connecting? Because बीच का nuclear membrane is completely what? Disappeared. Smera.
Yes, sir.
Understood?
Sir,
Chaliye. Yes.
sir, then where do the other organisms go and when the nuclear membrane disappears?
जोर से.
Sir, then where do the other organisms go when the nuclear membrane disappears?
Organelles, na?
Yes, sir, organelles in the cell.
They disappeared because कहीं पर क्या होता है बच्चों? Contained nuclear content keep on increasing. See why cell divides. Basic concept ये था that I think मैंने आपको बता दिया होगा. एक basic concept ये है.
Yes, sir. Ki.
This keep on pressurizing and this keep on expanding. This keep on pressurizing, this keep on expanding. So the material in between will embedded in that protoplasm, nucleoplasm and cytoplasm. Winners. Understood?
Yes, sir.
Yes, sir.
This is not a flat structure, no, my dear. This is not a flat structure. This is a complete like a ball of watermelon. उसमें अंदर embedded हो जाता है. Understood? कौन पूछा था?
Sir, Manoli.
Manoli madam, समझा क्या?
Yes, sir.
Perfect. So at the next moment, when we comes to anaphase, since these poles start shortening of fibers, chromosomal fibers. See, इसमें हुआ था expansion of fibers, expansion of spindle fibers. इसीलिए एक और point याद आ गया अभी. इसीलिए the charge on the spindle fiber is positive towards equator and is negative towards pole. Since it is expanding, it is positive. So पूछा जाता है what is the polarity towards equator? It is positive. It is positive. And towards pole, it is negative. Towards pole, it is negative. Towards center, it is positive. Because यहाँ पर एक तरफ लग रहा है pushing force और एक तरफ लग रहा है pulling force. So pulling is that negative pressure. So in anaphase, The characteristic feature is, इसका मैंने बताया 3 फीचर आपको बताया, इसका मैंने बताया इक्वेटर प्लेट, 3 फाइबर्स बताए, पॉजिटिव नेगेटिव मैंने बताया. Now it comes to anaphase. The characteristic feature of anaphase is chromatid starts separating. देखिए मैं बार-बार why I'm emphasizing the word. Chromosomes are not separating. Though you arrange only chromosomes on the equator. You arrange only chromosome on the equator. But now what you are separating is chromatid. That is what we were expecting. That is what we were expecting. But if there is a chromosome, ध्यान रखो कितना मजेदार स्टेटमेंट है ये. If this was one, मैंने आपसे कहा ये अभी भी एक है, ये इक्वेटर पर आ गया है, ये इधर and start thinning. तू तेरी सेल बना ले, मैं अपनी सेल बना ले. कुछ समझे मिश्रा जी?
यस सर.
प्राप्ति, प्रणव, पार्थ, नमिशा.
Yes, sir.
Yes.
Yes.
So what word
Yes,
I'm
sir.
using? Two state. मैंने हर किसी के दो एकदम I should say characterized feature मैंने आपको बताया भाई. इसका feature is now shortening, not condensing. Shortening of spindle fiber. यहाँ पर ये wall रखी है, ऐसा मैंने हाथ लगा दिया, touch हो गया. Now what I'm doing? I'm not condensing this spindle fiber or chromosomal fiber I should say. I'm shortening of this fiber. Getting my point?
Yes.
It is not condensing at all. This fiber is not condensing. This is shortening and moving towards negative pole, and it becomes like this. And one chromatid is here, and one chromatid is here. One chromatid is here, and one chromatid is here. Understood, बच्चों?
Yes.
Understood?
Yes, sir.
Yes, sir.
And therefore you can easily see different shapes. And this is the identification. If we tries to observe any mitotic division, probably you had conducted this experiment in your laboratory. I don't know whether in this COVID or online situation, whether you have conducted this experiment or not. Onion root tip. Onion की जो root tip होती है, उसका division का, उसका mitotic divisions देखने का तरीका होता है. तो इसमें English alphabet के different shapes we identified, like J, like L, like V, like I. Why these shapes appears? ये भी बताऊं. Because there are different types of chromosomes, like one which is exactly in the center. This is metacentric chromosome. So when this will separate, what shape will you expect?
V.
What expect? अरे भाई इधर से ये आएगा और इधर से ये खींचा जाएगा, तो V शेप आएगा ना? Yes or no?
Yes, sir.
Yes, sir.
Yes, sir.
यदि मैं आपसे कहूं कि if it is not metacentric, it is submetacentric. Submetacentric is like this. So when someone will pull from these two poles, it will appears like L shape, English alphabet L shape. Yes or no?
Yes, sir.
Perfect. If it is telocentric chromosome, then like this. So
J shape.
when you will stretch this, that will be J shape. Yes or no?
Yes, sir.
Agree? So different alphabetical should get different shapes of English alphabet. That is the identification of this anaphase. Technical बात करते हैं. Condensation of chromatin material, disappearance of nuclear membrane, disappearance of some organelles. All the chromosomes comes to the equator plate. All 46 बैटरी बिल्कुल 10% बच गया बच्चों, स्टिल ठीक है, नो प्रॉब्लम. About to finish. Chromosomes comes on the equator plate. Single metaphasic plate is there. The positive side towards equator, the negative side towards pole. Anaphase. Spindle fiber starts shortening, embedded into pole. Different shapes appears. And what separate chromosome or chromatid?
Chromatid.
Chromatid.
And finally, due to this constriction, the pressure creates on the cytoplasm. And in telophase, it completely embedded, and start again the thinning of this chromatin material. And so that a cell furrow will be there in animal cell. This is telophase. Telo means terminal phase. Pro means first phase. Telo means terminal phase, which is followed by which phase? Cytokinesis. Perfect. So due to this constriction, centripetal force, बताऊंगा ये सब लिखेंगे अपन डिटेल में, but concept पकड़ में आना चाहिए. Again it becomes thin. So for a moment, for a very short moment, you will see some statement in whatever the reference book we are using. For that moment only, very short moment, each chromatid has its own centromere. Did you understand this statement? For a very short moment, each chromatid has its own centromere. Agree or not?
Yes.
Yes, sir.
Why I'm telling this? Why? What we were separating? Tell me. What we were separating? Chromatids. तो अभी तो भैया मैंने जब क्रोमोसोम जो मुझे दिखा, जो दिखता ही नहीं कभी, वो तो दिखा इसलिए क्योंकि कंटेंट डबल हो गया. वो जो क्रोमोसोम दिखा, वेयर आई एम पिकिंग दिस पेन मार्कर, जहां से मैंने होल्ड किया हुआ इसको, दैट पॉइंट इज centromere. That is only one centromere. Yes or no? Yes or no?
Yes. Yes,
So
sir.
this centromere, for a moment in anaphase, it separate as a chromatid. So for that moment only, since it now going to become a one more chromosome or chromatid, so इसका अपना centromere होगा. Yes or no?
Yes, sir.
Yes, sir.
Agree? वैसे सेंट्रोमेयर दिखता ही तब है बच्चों जब वो क्रोमोसोम होता है, बट यहाँ लिखते हैं कि व्हेन क्रोमेटिड सेपरेट्स इंडिविजुअली, कहीं आपको पढ़ने में दिया जाए तो डोंट टेक इट अदरवाइज, उसको बिल्कुल समझने की कोशिश करें कि थोड़ी देर के लिए उनके अपने खुद के सेंट्रोमियर हो सकते हैं, क्योंकि सेंट्रोमियर ही स्प्लिट होता है. एक्चुअली व्हाट स्प्लिट्स
Centromere.
Centromere.
बिकॉज काइनेटोकोर तो दोनों का अपना अपना काइनेटोकोर तो था ही. सो सेंट्रोमियर स्प्लिट, और जब यहाँ लिखा होगा टेक्निकली सेंट्रोमियर स्प्लिट, तो मतलब ही यार दो सेंट्रोमियर बन रहे हो. तो फॉर अ मोमेंट अगेन इट बिकम्स थिन एंड फिर सब डिसअपियर. कुछ जरूरत नहीं, क्योंकि थिन में सेंट्रोमियर दिखेगा क्यों? सेंट्रोमियर तो तब दिखेगा जब वो क्रोमोसोम दिखेगा, और वो तब दिखेगा व्हेन सेल एंटर्स इनटू द न्यू डिवीजन. विनर्स, अंडरस्टूड और नॉट?
Yes, sir.
Yes, sir.
Perfect. So should I give you some assignment? Should I?
Yes, sir.
Yes, sir.
I'm sharing an image from a reference book. मैं इमेज को शेयर कर देता हूं आपको, उसको देखिए. एंड आई नीड ओनली 3 points of each phase. Is it possible?
Yes, sir.
Yes, sir.
Perfect.
Yes, sir.
I expect because मैंने बहुत अच्छे से आपको समझाया है, तो मैं एक्सपेक्ट भी करूंगा और एक इमेज में आपसे शेयर. जस्ट मिनट, आई ट्राई. 8% बैटरी बची है. जस्ट मिनट, आई ट्राई. हो जाएगा तो हो जाएगा.
सर वो क्वेश्चन है क्या?
हाँ, कोई बोला कुछ?
सर वो क्वेश्चन है क्या जो आप भेजने वाले हो?
आवाज नहीं है. कौन बोला?
सर वो क्वेश्चन है क्या जो आप भेजने वाले हो असाइनमेंट में?
नहीं, क्वेश्चन नहीं है. मैं इमेज आपसे शेयर कर रहा हूं दोस्तों. जस्ट मिनट. यस. काइजमेटा अभी नहीं बनेगा बेटा. किसी ने सवाल भेजा क्या काइजमेटा का? Please check this. Can you see the screen?
Yes, sir.
Okay.
Yes.
So just go through what you are seeing there. Simple, starting with the interface, सब thread है, chromosomes बनने लगे. Please check कीजिए और किसी भी स्टेज में डाउट आ रहा है तो तुरंत बोलिए मुझे अभी. I'm trying to go for a video. Please check and tell me कि सर ये स्टेज नहीं समझ में आई. अर्ली प्रोफेज, मिडिल प्रोफेज. देखिए सुमेरा, सेंट्रियोल की मूवमेंट देख रही हैं आप? थर्ड वाले में, थर्ड सर्कल.
Yes, sir.
बिकॉज वो न्यूक्लियर मेम्ब्रेन डिसअपियर हो गई तो वो कॉर्नर पर पोल पर आ गया. 3 टाइप के फाइबर्स भी दिख रहे हैं. देखिए कंटिन्यूअस, डिसकंटिन्यूअस एंड क्रोमोसोमल. गॉट इट?
Yes, sir.
अर्ली एनाफेज यू कैन सी द डिफरेंट शेप. यहाँ तो सारे वी शेप, अल्फाबेट बी फॉर विक्ट्री दिखाए गए. फिर लेट एनाफेज में वो शॉर्टनिंग हो गई उसकी, एंड दैट एम्बेडेड इनटू दिस एंड फॉलोड बाय साइटोकाइनेसिस. अग्रीड?
Yes, sir.
अब मैं एक शानदार चीज आपको दिखाता हूँ. यदि बैटरी मुझे सपोर्ट कर दे तो आई कैन शो यू परफेक्ट वन.
Excuse me, sir.
एक्सक्यूज.
Sir, what is interzonal fiber in that picture?
जस्ट इज इट ऑडिबल विनर्स? इज इट ऑडिबल?
Yes.
अरे आवाज आ रही है बच्चों?
No, sir. Not properly.
No, sir.
वीडियो की आवाज नहीं आ रही है?
No, sir.
Sir, आवाज नहीं आ रही लेकिन दिख रही है.
अभी
No, sir.
क्या करूं इसको मैं? चलिए देखिए कम से कम.
और आप सबटाइटल ऑन कर दीजिए तो वो क्या बोल रहे
हाँ?
हैं. सबटाइटल ऑन कर दीजिए तो वो क्या बोल रहे हैं तो समझ में आएगा. तो ऑडियो क्लियर नहीं है तो उससे पता चल जाएगा.
क्या करूं मैं?
सबटाइटल
सर
ऑन कर
सबटाइटल वो जो सीसी है ना
वो
ऊपर.
सीसी को क्लिक करिए.
इसी को क्लिक करूं. अब ठीक है?
Yes, sir.
हाँ.
Yes,
स्टार्ट
sir.
करूं? different fibers.
Sister chromatids are held at the center of the core and the surface. The kinetochore holds the chromosome and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores and the daughter chromosome along the divided centromere to the divided poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole, and the chromatid starts decoying. The nuclei form along with nuclei. This triggers the recognition of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage appears in the plasma membrane, which divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, cell formation is triggered at the center of the cell by the formation of the cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei. Mitosis involves four key phases: prophase, metaphase, anaphase, and telophase.
Got it, doctors? The winners?
Yes, sir.
Yes, sir.
Yes, sir.
I hope जो आपने पढ़ा उसको वीडियो देख लिया तो एकदम से दिमाग में बैठ जाती ना चीजें.
Yes, sir.
Yes, sir.
Perfect.
Yes, sir.
I hope असाइनमेंट आप पूरा करेंगे बच्चों. बैटरी भी जवाब दे रही है, 3% बची है बैटरी. आप इसका असाइनमेंट आपको लिखना है साइटोकाइनेसिस, मतलब फेजेस के 3 points each phase, and cytokinesis both plant and animal. See you in the next class, that is with meiosis. Right? Any doubt in today's session?
No, sir.
Perfect.
सर
All
आप
the
वो
best.
इमेज भेजने वाले हो ना ग्रुप पे?
कौन सी?
रेफरेंस
Yes.
बुक की.
मैडम अभी लीजिए. आपने बोला, कौन बोला?
सर मनोली.
जी मनोली मैडम, ये लीजिए. अभी तुरंत ही आपके विनर बैच पर ये पहुंच गया. इससे पहले कि मैं बंद करूं, प्लीज चेक एंड कंफर्म. डिड यू रिसीव इट? हैव यू
Yes,
रिसीव्ड
sir.
इट? Received?
Yes, sir.
Yes, sir.
Perfect. Any more doubt? Oh, Mishra, understood?
Yes, sir.
Chaliye, all the best. See you in the very next class.